Welcome, everyone. Thank you very much for joining us for our Ecological Continuity Trust webinar series. Just a couple of quick housekeeping reminders before we start. If you could please keep your camera turned off during the seminar, that just helps save bandwidth. And also, if you can remain muted as well, that would be really helpful. If you have any questions as we're going through, please do put them in chat, and Ben will go through questions at the end and may ask you to unmute and comment or ask questions. So I'm very pleased to introduce Sara. Sara has recently completed her PhD in the biology department at the University of Oxford. She's an interdisciplinary plant ecologist with a background in environmental scientists, and she has broad interests from plant traits, grassland ecology, plant-plant interactions to botanical education and outreach in the context of global environmental change. Her field-based research focuses on trying to understand environmental change impacts on vegetation functioning across space and time. And Sara has been working on the RainDrop experiment, which is an experiment that ECT played an important role in establishing. So very pleased to have you with us, Sara. Over to you.
Great. Hello, everyone. So yeah, thank you very much for inviting me to speak today. I'm going to be speaking about my work at RainDrop and collaborations with collaborators as well. And just to set expectations, I have quite a few slides. I have 95 slides, but they're mostly pictures. And I'll do my best to keep to time. So start with the first slide. So let's see, this should play. This is RainDrop. So this is like a drone shot of RainDrop. We haven't been there before. It's a really beautiful site just outside of Oxford, and it's on a hill. It's about 160 meters above sea level, and it's a calcareous grassland. And these are the shelters, which I'll explain more in just a moment. And let me just pause that one. So I also want to acknowledge that the research at RainDrop is really vast, and what I'll speak about today sits in a wide context of other research. So there's been research done on plant pollination interactions, on some below ground things, to trying to automate vegetation monitoring, even looking at soil microbiota, to plant traits and community vegetation responses and herbivores. So there's a great deal that's going on at RainDrop. So I just want to acknowledge that there's a lot of exciting research. So I'm just going to focus on things that I did during my PhD and with some collaborators as well. And if you're interested, you can look on the ECT's website, and it has a sort of bibliography of all the publications and research at RainDrop. So you can just use that QR code if you're interested. So the RainDrop plant story. So I've organized my talk into sort of 4-ish parts, and I'll be using these symbols throughout. And the context of RainDrop, I'll give a little bit about history of the site and sort of key features. And then I'll talk about some of the research approaches that I carried out during my PhD. And I'll share some sort of highlights and sort of key results of the research, and also thinking about sort of future directions and potential avenues to look at in the future. So in terms of the RainDrop context, as I said before, it's at Wytham Woods, just outside the centre of Oxford. And thinking sort of back, like why was RainDrop sort of set up? If we're thinking about grasslands in general, especially like in the UK, we've lost 97% of our wildflower meadows since the 1930s. And if we're thinking specifically about calcareous grasslands in Britain, it's estimated that we've lost, well, there's been a decrease in almost 40% of grassland calcareous sites between 1960 and 2013. And these calcareous grasslands are under threat from multiple angles, from land use change and management change, agriculture expansion, invasive species, urbanization, and also climate change related threats such as drought, which is what I'll be talking about. And just so we're on the same sort of page, when I talk about drought, I'm thinking from an ecological plant perspective, and that is basically a deficit in water input relative to the normal levels or input relative to what the plant needs. And droughts can have various consequences for plants, increasing the stress and mortality of plants, for example. And we also tend to think of just the amount of rainfall in terms of drought, but actually there's other important dimensions of drought, such as frequency and how long that drought lasts, and also legacy effects of the sort of time aspect of droughts. So thinking in terms of responses to drought, if we think across sort of organizational level, so here I've got from tissues and cells all the way to sort of ecosystems, there are different responses, and they tend to be sort of hierarchical. And if we're thinking about sort of the drought dimension, which will be along the top as I go through the slides, when you if sort of we can have, before a drought happens, there are some like typically sort of annual plants have a fast life cycle that escape drought, so they do all their living and growing and reproducing before a drought hits. But if there's sort of a moderate drought, then you have these sort of responses at sort of different levels. So you can get shifts in productivity at the ecosystem level, and then in populations you get an increased mortality. As we go down, you get differences in root to shoot ratio, for example. And if we're going right to sort of tissues and cell level, you get a decrease in stomatal conductance. So these are just some examples of some of the symptoms of moderate drought stress. And if we go really sort of severe, then the impacts can lead to decrease in sort of carbon storage capacity if you're thinking sort of ecosystem level responses. If we're thinking sort of individual level, you can get dehydration tolerance or dormancy. And if we're thinking sort of organ and sort of tissue cell level, you can get early leaf senescence and reducing photosynthesis as well. And then post-drought, there's also how quickly these the vegetation sort of responds and bounces back to the prior state. And grasslands in particular are particularly sensitive to drought compared to sort of forested ecosystems. Generally it's thought because of the lower water use efficiency. And it's becoming increasingly a problem in sort of Europe and in the UK, thinking about droughts. I'm not sure if you remember, was it 2 years ago now, that we had a very severe drought, and I took this picture of a yarrow, and it has very sort of deep roots, and that was the only thing that was sort of like surviving in this green space. So we're going to get sort of more frequent droughts, so it's important to sort of study these impacts and see which plant species respond and in what ways. So that is the context of sort of RainDrop, sort of why we're interested in those sort of questions. And just sort of to contextualize it again, this is where RainDrop is. And if you were 160 million years ago there, it'd be much toastier and warmer because it was shallow tropical sea and it's full of corals. And this site is littered with a lot of these stones. And during the 1940s and 1980s, a lot of the land was converted to arable crops, and that was abandoned because it wasn't very successful. And then it was then allowed to recover naturally. with natural colonisation. And interestingly, also in the 90s, there was a climate change experiment at the Upper Seas for about 5 years, looking at the effect of drought and winter warming. And they generally found that deeper rooted species and plants with faster sort of life cycles are more tolerant to the drought. That's kind of just the context. So there has been an experiment there before. And then since 2016, the RainDrop was set up in collaboration with Oxfordshire University, Open University, ECT, and the Patsy Wood Trust. And there are 4 treatments. So there's the control, where there's procedural control and ambient control, which has 100% of incoming rainfall. There's the drought, which is 50% less of incoming rainfall, and then irrigated is 150%. And they're arranged in 20 plots, and there's 5 blocks, and it's a randomized replicated design. In terms of management, the treatments only apply from April to sort of very early October. And then the treatment is off, so the shelters kind of move around, so you can implement the drought. And in terms of cutting, there's the mowing with biomass removal in early July and end of September. And also this site is part of the DroughtNet network, which is a consortium of various drought experiments across the world. So in terms of the species there's 109 species there, and lots of lovely grass species, parasitic plants, and lots of orchids as well. So these are just some pictures that I've taken over the years. Right, so that's enough of the context. And in terms of research approaches, I'll be talking about 3 main sort of projects. I sort of describe this as the RainDrop stories through space and time. So we're thinking about sort of organisation from community level all the way to organs. And we have sort of the community project study, which is looking at species composition and functioning over time, and that involved vegetation surveys and biomass collection. And then the demographic study, which was over 3 years, which focused on a particular dominant grass species. And then a trait study, which was for 1 year. So I'll go through each project sort of in turn. So the first one is looking at the community level. So the main question was, what are the temporal dynamics in the structure and functioning of this grassland community? So that involved looking at using percentage cover, so with 1 metre quadrats, and those were done at the peak of the growing season across the treatments, and I'll present data from 2016 to 2021, although it's still ongoing. And in terms of functioning, that was looking at above ground annual net primary productivity, and that was looking at using biomass, and that was taken from 1 metre by 0.25 metre strips, and that was looking at the community level biomass and then split into functional groups with forbs, graminoids and legumes. And that involved lots of sorting, and it was really like a huge, lots of people sort of involved in sorting this because it's a lot of painstaking work to sort into functional groups. And you can see or hear the biomass bags ready to be put in the oven for drying. So in terms of results, unsurprisingly, graminoids are the dominant functional group by biomass, and they represented about 63% or 64% of the mean total annual net primary productivity, and sort of smaller percentages for legumes and forbs. And if we're thinking here about productivity split across treatments, so we have here the treatments along bottom, so we've got ambient control, procedural control, drought and irrigated, and then we've got productivity up on the y-axis. You could see that drought reduces the mean productivity roughly by about 25%. And similar responses in sort of other grasslands as well, so it's not totally unexpected. It could potentially be linked to sort of changes in nutrient cycling. So there was a study done at the Upper Seeds experiment, so during the 90s, where they looked at soil nitrogen mineralization rates, and they saw that during the summertime, the drought inhibited nitrogen, mineralization, but then it increased during the autumn and winter time. So there's sort of seasonality to that, and that can also impact. So these microbial sort of dynamics and decomposition affect then the nutrient release and so forth. So it could be sort of linked there. And then interestingly, there was sort of a muted response with irrigated plots. So it seemed to behave quite similarly to the controls. And we think it's probably likely due to either the amount of water, addition of water. So it was 150% of rainfall levels. Maybe that wasn't enough to elicit a response, or it could be to do with efficacy issues with the irrigation treatment potentially. And looking at comparing the ambient procedural controls, they have a similar response, which is good because it shows that the rainout shelters, the actual structure themselves is not impacting the vegetation responses. And it agrees with earlier studies by Melanie Stone, who's a PhD student before, who did some research on community responses. So that's good. Thinking about, again, productivity, which functional groups were driving that reduction? It was graminoids and legumes that are contributing the most to that reduction in productivity. And it could be to do with sort of grasses on the whole tend to have shallower sort of rooting depth, which make them more, susceptible sort of to drought stress, and they'll be contributing less to the overall biomass. And with legumes, they generally have higher water requirements because of their bacterial mutualism, so they could have been under more stress. So that's possible mechanisms for that. In terms of temporal stability and productivity, with drought it was reduced. That was measured with the coefficient of variation. So if you get a high number, then it means you got a decrease in stability. And the drought had a much greater coefficient of variation. So it's much more unstable. And you can see it here in this graph. And this sort of agrees with work in a different site at the Buxton experiment, where they were looking at seed banks, and they saw that had a similar dynamic there as well. We're now going to composition. We can see that there isn't really a difference here. We're looking at different measures of diversity. So we've got species richness, Shannon-Weiner index, and Simpson's index along the side. And you can see they're all behaving pretty similarly across the treatments. And this is resistant despite declines in sort of grasses and legumes. This might indicate sort of possible lag effects. Many of these species are quite long-lived. Long-lived perennials. So it could be potentially maybe like a warning signal in the future when there is that mortality and then turnover of species. But at the moment it seems stable. And what we did find is that there's treatment-independent shifts in community composition and richness. And so, as I said before, there was 109 plant species recorded. Mean number of species was 24. And there was an increase in about 7 species between the start of recording and the end, so between 2016 and 2021. And that's shown quite nicely here, as you can see. There's an increase, but there isn't really a clear pattern in species links in the functional groups there. So it's a bit unclear which species are sort of driving these shifts. But I think this sort of shows the sort of successional change of the grassland. So to summarize very briefly, the community, vegetation study. So species diversity and composition showed resistance to precipitation. The drought reduced the productivity and temporal stability, mostly driven by graminoids and legumes, as indications of possible lagged responses. Now moving down in scale, looking at the population individual level. Here I'm focusing on the bunchgrass Brachypodium sylvaticum, and I was interested in looking at various sort of demographic measures related to survival and vegetative and reproductive components, and also thinking about biotic interactions. So that involved doing a demographic census. I tagged 138 individuals and involved a lot of image processing of the plant recordings. And in terms of biotic interactions, that we're looking at the presence of ants, rabbits, species richness in sort of the neighborhoods, and also legume species. And just to sort of situate how the tagging worked, I sort of just tagged adult individuals in 2019, and then I made a sort of neighborhood which was 0.3 meters in radius, and that was kind of like the effect zone. A little bit about Brachypodium sylvaticum, if you're not familiar with the species. It's a perennial species. It's a woodland edge. It's found sort of woodland edge and scrub. Tends to favour calcareous neutral soils. It can grow pretty high, about 1 metre. I didn't find any 1 metre ones at RainDrop, but I have seen them in other places. It tends to be one of the later flowering plants around July and August, and it is wind and self-pollinated. And the reason why I chose this species that for my demographic study is that it was highly abundant at RainDrop, and it wasn't clonal, and it was easy to tell like what an individual was. And what I mean by individual in this case is the actual bunch itself. And also in the literature, it's classed as a model organism for various genetic studies into, for perennial crops. And it's also invasive in North America, so there's some interesting demographic studies done there. These measurements that I did, so I did non-reproductive height, reproductive height, looked at the number of leaves, number of flowering spikes, length of inflorescence, and the canopy area and basal area. And I also looked at survival and reproduction. And some of the recordings were done on these acetate sheets. So basically just with a Sharpie drawing around them. And this is kind of the setup, what it looks like. And on those same sheets, I also recorded biotic interactions. So within the neighborhoods, I just noted down the species that were present there. And I made a note whether there was a presence absence of rabbit activity or sort of anthills. And these were listed as covariates. So this is what it actually looks like. And these are the sheets, and this is the sort of setup. So very low tech. In terms of processing the sheets, that was probably the hardest part of the PhD process. So this is me recording out in the field, and then I had to take a picture of them. And there's almost like 400 sheets of them over the years, and they're very, very detailed, as you can see here. So you can see the black splodge is the Brachypodium sylvaticum, and then all the little blue splodges are the neighbouring species, and then the circle is the sort of neighbourhood area. And then it involved a lot of trying to extract the plant size data using image segmentation. This one is probably my favorite because it looks like Patrick Star from SpongeBob. But yeah, it was very, very challenging, and I was very thankful for some sort of help from computer scientists to try and automate this process. And I wrote a little article about the process there if you're interested. And the following results that I'll show will be pooled because, as I said in the community study, the procedural control and the ambient control had very sort of similar results. So they will be pooled. So when you see control, it means the pooled results. So what was really interesting with the Brachypodium sylvaticum is it had very high survival rates regardless of treatment. So 93.4% of them survived into the growing season, which was I was a bit surprised, and I realised that, oh, actually there's a lot more work for me because I have to go out the next year and like record them. And I suppose on the whole it's not surprising in the sense that there's been some long-term studies, I think in Sweden, which shows that they can live for up to like 20 years. So this study is about sort of 10% of their lifespan. In terms of reproduction rates, very low, so only 8.7% of them, so only 12 of them reproduced, and it was a range between like 1 to 6 new recruits. And there was 23 new recruits in the study period, and most of them were found in 2020. I did want to do, or I originally set out to do some more analysis on the sort of seedling dynamics, but it just wasn't enough for sample size. In terms of looking at the effect of treatment as a whole, I've just shown here, so simplified ANOVA table, showing the fixed effects of the models. So on top, we've got the 7 demographic sort of measures, and then we've got the sort of fixed effects, treatment year, and the interaction. And as you can see here, for the plant height, so non-reproductive height and reproductive height, it was significant. And then also year was important. And this is shown here, looking at the interannual variability across the measures. So each one of these graphs shows year along the bottom. So we have 2019, 2020, and 2021. And then we've got each of the demographic measures. Hopefully you can see that. So we're just looking at the general patterns here across years. So you can see that there's a lot of sort of change between years. And if we're focusing on plant height, so both non-reproductive and reproductive height, with drought here, so that's the yellow, orangey effect, and with the sort of bars there, you can see there's a really great reduction, and with drought. And irrigated and control seem to have similar-ish responses. And I thought what was very interesting for reproductive height is that there was sort of a possible sort of drought-like effect. It took a while to recover. So it took an extra year longer to recover than it did for non-reproductive height. So I'm wondering also if this is partly sort of a slow recovery from the 2018 severe drought. So I've only got data from 2019, but if you remember from 2018, it was incredibly dry. And that's when I first saw the experiment. I was like, oh wow, this is what have I signed up for? It's like completely dry. Is this place going to, you know, recover? It was all parched. So I'm wondering if that's kind of a kind of delayed effect there, so it didn't recover as quickly. And then thinking about the random effects now. So which effects were contributing the most to responses? Here we have each of the sort of demographic responses, and then we have block plot and residual for each of them of the terms, and then we have the standard deviation along the bottom. You can see that here's a large residual variation across all of them, and that could be probably a large part of that is individual level variation. So there will be, some measurement error, etc. But I don't expect that to be a large proportion. And then if we think about adding in sort of treatment and year, so if we use the sort of like Gelman and Over trick, where you put all the terms, so here block plot, treatment and year into your random, then you can kind of more easily compare. And again, here's sort of a large overlap in the sort of variance component. So that's kind of, interesting what's happening there. I'm not quite sure exactly why they're all like that. But yes. Okay, moving on to co-occurring species. So this is within the sort of neighbourhood. A total of 67 species were found in these neighbourhoods across the 3 years, and the mean number was 15. And in terms of the top 10 species, we have the sort of graminoids here, and some lovely legumes and forbs as well. So these species, and they remained largely stable across the years, which has similar results to the community results that I shared before. And if we're looking at the other responses as well of the sort of covariates, which I mentioned before, so that was looking at the effect of rabbits, ants, legumes, and then the sort of total number of species. The effects were sort of largely consistent across all the different measures. There was a slight sort of negative effect of rabbits and ants, and slight positive with the presence of legumes, but it still is a large overlap, so I don't want to like over-interpret that. So the demographic study, to summarize, there was high survival, low reproduction. You could characterize the species as a conservative with its resource use and stress tolerant. Individual level variation important, but there is a large overlap. And as I said before, biotic interactions were similar across the variables, and indication of possible lag effects. And oops, it's frozen again. Now onto the final one. Conscious that I'm going a little bit over time, but I will try and speed up. This is the last one, looking at traits, and this is looking at sort of individual and organ level. So here I was interested in looking at above ground trait responses and how they vary between and within Brachypodium sylvaticum individuals across the precipitation treatments. And it's important to note I used the survivors from the demographic study. So there's a slight survival bias there, so just to keep that in mind. So that involved various trait measurements during the peak growing season in 2022, and as I said before, it's the adults that survived into 2022, and also looking at sort of the trait space. And these are the traits that I measured. So I did leaf thickness, leaf area, leaf dry matter content, specific leaf area, stem specific density, and maximum height. And these can sort of give indications into drought responses. So for example, leaf area, you might tend to get sort of smaller leaf sizes with drought. With stem specific density, it gives you an indication of growth survival trade-off, and maximum height gives you an indication of competitive vigor and reproductive size. And with drought, they tend to get smaller stature. In terms of looking at the mean trait responses, it's sort of stable across the treatment, as you can see here. So we have, remember these pooled controls. So you have control, drought, and irrigators, and then you have the so for each what's the word? Sorry, variable. And as you can see, like the means, there isn't really a strong difference in them. But if we think about beyond the mean and looking at the sort of trait space, we can see here again for the same 6 traits at their density distributions here. You can see that there are differences beyond the mean, and we'll ignore stem-specific density with that strange trimodal distribution. I think there wasn't enough data for that, so it was acting a bit weird. I don't think there's anything biological going on there. And I think, so I'll highlight some key things that I noticed. There was a narrow distribution for leaf thickness across the treatments, and I had a very small, or comparatively small, coefficient of variation, so it was only like 5% compared to some other traits. I think stem-specific density was about 45, so it was very, very variable. And this was independent of treatment. So I'm wondering if for leaf thickness it's a more of a conservative trait in this species, and it doesn't really, you don't get sort of the phenotypic plasticity. Another interesting one was a wider distribution for specific leaf area. Usually when you have some stress, it usually results in a sort of narrowing of the trait distribution. So it's quite interesting that drought actually had a very wide dispersion, and it's not what I was expecting. And I'm wondering if it's to do maybe a slight issue with sort of measuring a specific leaf area, because it involves measuring the leaf area. And I saw a lot of presence of leaf rolling in the drought plots. So you can sort of see in this image here where the leaves kind of go inwards to kind of reduce the water loss, basically. And it can be sort of harder to sort of tape it down when you take the measurement for leaf area. And then the other thing that was really interesting, which I was not expecting, is that in the drought plots, I tagged them and I went to the same place each year. I noticed quite a few of them, of the individual bunches in the drought plots, were actually moving towards the outside of the plots, so the center of the bunch. So I would go in and put my sheet, and I'd be like, "Oh, you've moved from last year," because it was tagged. So I'd go to the same place, but I noticed it moved. So I'm wondering if that's also a response that I didn't, I observed, but I didn't directly measure, that could be contributing to the response. So if we're seeing more stable responses in the traits that I measured, it could be reflected in other traits that are more plastic. So I thought that was really interesting as well. In terms of looking at the source of variation, again, it wasn't very, there was no clear sort of main driver in sort of the responses. And this is looking at sort of block, subplot, and individual level. There's a large degree of overlap there. And again, when you include treatment in there, there's a large degree of overlap between the different levels of organisation. Possibly it could be to do with, because it's a small study area, it could be there's similar sort of biotic, abiotic processes happening at the site, but I'm not entirely sure the reason for this. So yeah, just to summarize, the trait study, there were sort of minor adjustments in leaf anatomy with a reduction in leaf size and evidence of leaf rolling, which was helping to sort of maintain sort of optimal functioning. There was no clear source of variation in driving the responses, as the large degree of overlap. And then with leaf thickness, possibly more phenotypically, sorry, less plastic. And yeah. So to sort of summarize, looking at the RainDrop story through space and time, I've looked at the community level, and then sort of population, more meta-population, individual level, and then sort of individual and organ level, different times from 6 years, 3 to 1 for the trait study. And overall, the community level composition is resistant to the drought, but there is reduced productivity and temporal stability, especially driven by grasses and legumes. For Brachypodium sylvaticum, it shows that it's drought tolerant and it has a high survival and low reproduction. And looking at the traits, it seems to show that it has optimal functioning, where it tends to have quite stable trait responses with only sort of minor adjustments with some of the leaf traits. In terms of potential next chapter for the RainDrop story, there's various points where things can be extended, and things that I found really interesting. For me, particularly, was like looking at would be good to look at below ground traits, which I intended to do, but it's a very hard site to work out, so I tried to put the shovel in, and it just didn't go in. I think because of all the ploughing from like previous management makes it very hard to work in the site. So you'd probably have to do experiment in greenhouses if you want to look at root traits. It would be interesting also to look at sort of genetic pathways to see, especially for the trait responses, if these are adaptive versus sort of reactive, and also genotyping would be helpful to sort of decompose the source of the variation. And with sort of all the sort of studies, thinking sort of longer term, because especially with the traits, that was just in one growing season, so it's like a snapshot. So it'd be interesting to see, do we see these responses repeatedly over the years? And for the demographic study, it'd be interesting again to have longer term monitoring, especially for the species, because it has such a long lifespan. And to characterize a bit more the sort of biotic interactions more formally. And also, it'd be interesting to look at thinking specifically about drought, to maybe make the drought treatments more extreme, because maybe for the Brachypodium sylvaticum, it wasn't quite at its tolerance sort of limits. So maybe thinking about, remember the different sort of drought dimensions, so thinking about maybe doing it earlier in the growing season, because grasses can be a bit sensitive if they do most of their growing in sort of springtime or sort of late winter. That would be interesting. And also thinking about, as I said sort of right at the beginning, that grasslands have multiple threats, it's not just drought, so having multiple stressor experiments would be interesting also. And then to sort of end it there, I suppose, to summarize, I think to understand sort of the plant stories and the sort of characters going on in the community in a sort of changing climate, and multiple perspectives across sort of space, time, and organisation are needed. And with that, I've got lots of people to thank for helping me with field work, and just having conversations and bouncing ideas and so forth. So yeah, I'm happy to take any questions.